Hello, my name is Connor Hill and I'm a PhD student at the University of Pennsylvania in Dr. Alessandro Gardini's lab. I am interested in using data science to make discoveries in transcriptional regulation in human diseases such as cancer. My current project, which I am presenting today, is studying a rare tumor type called Solitary Fibrous Tumors, or SFTs. They are found throughout the body around connective tissue. Most tumors are benign and can be easily removed through surgery, but 10 to 20% of them become malignant, and unfortunately there are no effective treatments for these cases. It is not known how these tumors develop or what makes them become malignant. There are no recurrent mutations except for an intrachromosomal inversion that produces a gene fusion between the genes NAB2 and STAT6. This gene fusion is present in all SFTs and is unique to them. It is currently how these tumors are diagnosed in the clinic. However, it is unknown how the NAB2-STAT6 fusion protein functions and contributes to the development of SFTs. Both NAB2 and STAT6 normally function as transcriptional regulators, NAB2 through a close interaction with the EGR family of transcription factors, which are important in various developmental processes, particularly neuronal pathways, while STAT6 is a transcription factor in the JAK-STAT signaling pathway, which is involved in various immune processes. To study NAB2-STAT6's function, we developed an inducible expression system where we can induce expression of a flag tag, NAB2-STAT6, with the addition of doxycycline in U2OS cells. And as you can see here, by 12 hours by Western blot, we were able to get pretty strong expression of NAB2-STAT6. And after 2 days of expression of NAB2-STAT6 through RNA-seq, we begin to see widespread gene expression changes. And we then wanted to see what pathways were being upregulated, so we did Go enrichment analysis and saw that the top pathways being upregulated were those involved in neuronal development, and that we then wanted to see which transcription factor binding sites are found in these genes, and found that the top binding motif found in these genes was the EGR1 motifs, and these results suggest that NAB2-STAT6 is triggering an upregulation of neuronal EGR targets. To see how NAB2-STAT6 is doing this, we wanted to see NAB2-STAT6's binding along the genome using ChIP-seq, and we did this using the flag antibody for the tagged version of NAB2-STAT6 and identified about 1,400 NAB2-STAT6 peaks, as you can see with this heat map. We also did EGR1 ChIP-seq and found that these sites had significant EGR1 localization even before expression of NAB2-STAT6, and that the accessibility of these sites increased about twofold after localization of NAB2-STAT6. And finally, we did motif analysis and found EGR motifs at these sites, as well as gene set enrichment analysis, and found that the corresponding genes to these peaks became significantly more expressed when NAB2-STAT6 was being expressed. Finally, we wanted to see what proteins NAB2-STAT6 interacts with, and to do this we did IP mass spec for NAB2-STAT6 using antibodies for both NAB2 and STAT6. This plot on the right shows log2 IBAC values, a normalized measurement of protein abundance. So the most abundant proteins in both experiments are going to be here in the top right. And unsurprisingly, the top two proteins we pulled down were both NAB2 and STAT6. But interestingly, we also got NAB1, which is known to heterodimerize with NAB2, as well as EGR1 and other transcriptional regulators. And these results suggest that NAB2-STAT6 interacts with both NAB1 and EGR1 to function as a modulator of gene expression. We next wanted to see if similar gene expression changes occur in primary SFTs. So we were able to obtain FFPE sections from both SFTs as well as matching normal tissues. We obtained about 7 of these SFTs, extracted their RNA, performed exon-targeted sequencing, and as you can see here, SFTs undergo large-scale transcriptional changes, and just as we saw in our inducible system, we see upregulation of neuronal EGR targets. Finally, we wanted to see NAB2-STAT6's genomic localization in a primary SFT, and we were lucky enough to obtain a fresh primary SFT and dissociate it into single cells, perform ChIP-seq for both NAB2 and STAT6. And as you can see with this heat map, we identified about 700 NAB2-STAT6 peaks that had both significant NAB2 and STAT6 localization. These peaks also had significant RNA polymerase II localization, indicating that active transcription may be occurring at these peaks. These peaks had EGR motifs and also became, and their corresponding genes became highly expressed in SFTs. So in conclusion, we saw that NAB2-STAT6 increased the expression of a neuronal signature made up of EGR targets and localized to these EGR1 targets that then became much more expressed and much more accessible. And we then found that NAB2-STAT6 interacts with transcriptional regulators such as EGR1, and that in SFTs, neuronal EGR target genes also become highly expressed, and that in SFTs, NAB2-STAT6 localizes to EGR targets that become highly expressed. Thank you for listening to my presentation, and if you have any questions or would like to get in contact with me, my email and as well as my LinkedIn are below. Thank you.